Stanford University.
Thank you very much for the invitation to present here today. It looks like a great lineup, I agree. I'm very honored to be one of the speakers, and thank you for not saying all the other things that probably you could have said, although those were embarrassing enough. Actually, riding my bike every day, even in rain and snow, is just a statement about the traffic in Boston, actually. It's much worse to drive or even to take the T. At any rate, I'm going to not talk about the two topics I put in the title. In the title, I didn't pay enough attention to the timing, the amount of time I had for this talk, and I thought I could talk about development, which is really the topic that I care about the most, as well as nanobodies, a new technology that we've developed. And I realized with 35 minutes I could only cover one of those. So I decided to cover nanobody technology because I hoped that some of you would find that useful. It's perhaps of broader interest. So I will start with that. So I'll tell you what nanobodies are in a moment if you don't actually know, but I'd just like to acknowledge Jonathan Tang. So Jonathan was a graduate student in the lab, and then he stayed on for a year as a postdoc. And really, all of the ideas, all these great ideas that I think are behind what I'm about to tell you about, and the majority of the work were done by Jonathan. So this picture here is a structure of GFP, which is sort of the star of the show for the first part of the talk. So we use nanobodies in a variety of ways, and I'm going to tell you three stories. The first two use nanobodies directed against GFP. The first story I'll tell you is about using GFP and nanobodies to drive transcription inside cells. The second one is to use the same GFP nanobodies to drive Cre recombination. And the last is an attempt to expand the use of nanobodies to any intracellular cell type specific protein to drive many different types of activities. So first, the motivation for these studies is probably the same motivation that many of you have. If we want to manipulate cells in a complex tissue, we like to have genetic access to specific cell types. And so we work in the retina, and this line up here is the bipolar cells, a set of interneurons that we find in the retina. And we would like to understand how you can generate the diversity of cell types in the retina, or more generally in any complex tissue. And when Jonathan joined the lab, he wanted to study how do these different types of bipolar cells get generated and how do they distinguish themselves. But we couldn't find lines of mice. We like to manipulate specific cell types using lines of mice in which Cre or FLIP, two recombinases, are expressed in specific cell types. And when Jonathan started to look for those kind of lines of mice, he could not find them. But what he could find were lines of mice where GFP was expressed in subtypes of neurons, including, in this particular example, two types of bipolars. So Jonathan and I would sit and bemoan the fact that there weren't recombinase lines for our studies, and we kept coming across these beautiful GFP lines. Here's actually a picture of that. Most of these GFP lines use cis-regulatory regions to drive expression of GFP. And in fact, there's a very large collection of those that are called the GenSet Project, created by Nat Hines and his collaborators at Rockefeller. And you can just see some beautiful images of GFP-positive cells and subsets of neurons throughout the nervous system. Now, most of these are actually not cell type specific. They light up a specific subset, more than one type usually, but they're quite useful for many applications, as people have found. And again, they're created by these cis-regulatory regions. In the GenSet case, these are back transgenics. And we estimate from the GenSet as well as other people, there's quite a large collection, about 1,500 of such lines. So in looking at this, we realized this was an incredible resource in terms of giving us some type of access to specific cell types. But GFP in and of itself does not allow you to manipulate biological activities. So Jonathan asked himself, well, could we develop a way to use GFP itself to allow us to manipulate biological activities? And when he started reading about it, he realized that there was a particular point of access, and that was through nanobodies. So most of you know that antibodies, the type that we typically use in the lab, are 4 chains. The IgG types have 2 heavy chains, 2 light chains. And there's a variable region of the heavy and light chain that come together to define the epitope binding pocket. Well, camelids, which are camels, alpacas, llamas, as well as cartilaginous fish, have this 4-chain arrangement, but they've evolved another arrangement, which is just heavy chains. And the marvelous thing about these are that the heavy chain, the variable region of the heavy chain by itself will bind antigen. So you can then isolate just this domain from an immunized camelid and search for those that have the activity you want. Sorry, I think this is on auto advanced. Should have changed that. At any rate, this is a crystal structure of a nanobody for GFP, called GBP1, binding to GFP. So these small domains are only about 12 kD. You can express them inside cells. And when Jonathan was reading about nanobodies, he found that Rothbauer and Leonhardt at the LMU had created a collection of 7 nanobodies for GFP. So his idea then was to take pairs of those and to see if he could find pairs that could co-occupy inside a cell GFP. Now, this in of itself is not particularly useful, but his idea was to make fusion proteins and then introduce those 2 fusion proteins into a cell. And if GFP was present, it would bring those 2 fusion proteins into proximity so that they could create a novel activity. And this would be the way then to drive activities specifically in cells that express GFP. So he told me this idea. I thought it was great, but at the back of my mind, I realized, most of these great ideas don't work out in biology. There's always some glitch you can't anticipate. But nonetheless, Jonathan went ahead and he set it up first in vitro to look for transcription activities driven by GFP. And the idea is shown here. First, I should say, most of you probably are aware that transcription factors have been studied now for decades. And the finding is that they're modular proteins. You can have a domain of a transcription factor that activates transcription, another domain that might repress it, another domain that recognizes a specific DNA sequence. So this was a good starting point because we knew through fusion proteins that had been created with these modular domains, it was straightforward to create fusion proteins that had these activities. So he started there by taking a transcription activation domain, actually he used several different ones, and fusing it to one GBP, taking another GBP, fusing it to a DNA binding domain that would recognize a specific sequence, and we started with the Gal4 system from yeast, then introduced that sequence in form of a plasmid driving a gene of interest. We used TD tomato fluorescent red protein for many of our studies. And then the hope was that these 2 proteins would come together if GFP was present, bringing the activation domain close enough to then activate the gene. So this is a ternary system. 3 components are required in addition to GFP. And he started by looking at this in vitro using luciferase, a quick, easy-to-score readout. And he found that indeed the system was very robust at inducing luciferase. So this is now luciferase levels with different combinations of components. If you look at this bar, this is the bar representing the activity of luciferase when all components were present. If any one of the components was dropped out, you can see that we lost a huge amount of activity. This is actually an incredible induction of about 1,000-fold. So this worked well in vitro. He then wanted to see if he could make it inducible by small molecules. So GFP in the setting, for example, of a transgenic would give the spatial or cell type control. But being able to turn the system on then with a small molecule was the next goal. And so he checked to see if the TET activator or the TET repressor, which is activated by a small molecule called doxycycline, could be used. So he fused the DNA binding domain for TETR and made a plasmid that had its recognition sequence and asked whether he could turn the system on when all 3 components were present and the drug for activation was present. And again, in vitro, this worked very well. So if you look over here, this is the activation of luciferase when doxycycline was added and all the components were present. So again, a several 1,000-fold induction that's accomplished by the addition of drug. And these drugs can be added to animals. This has been used fairly widely to activate transcription. So we wanted then to see if this would work in vivo, and we do work on the retina. And this is our go-to system for testing anything. We're able to electroporate plasmids in fairly trivially into a postnatal day 0 mouse. And here we were adding 5 plasmids at once. The retina takes up quite a few plasmids and allows us to do these kind of experiments quite easily. So we had 1 plasmid encoding GFP to activate the system. We had this TD tomato readout to see if the system was working. We had plasmids encoding the fusion proteins, and we had an electroporation control plasmid. We introduced this into the retina in vivo, and then we let it go anywhere from a few days to a few months, and then we look at sections to see if the GFP-positive cells have turned on tomato. So we can ask several questions. One is just, is the system working? Is it dependent on GFP? We then worked with Botan Raska's lab to see if we could do physiology with the system. We asked whether the gene that we turn on is not tomato, but Cre, and if so, would the Cre accomplish recombination? And then we wanted to look further at physiology. So by doing these introductions with electroporation and then looking at sections, we could see the system was very tight. So these are now sections, and this is this series of sections from an animal that received GFP and this is a control with no GFP present. So you can see when GFP was present, TD tomato, our readout, indeed did turn on. So this is very pretty efficient to turn on, as a matter of fact, with no GFP, almost no TD tomato. But Jonathan's very honest and told me I had to show this section because there is one background cell. So when we do quantify, it's well below 1%. So without GFP, these components don't come together spontaneously to create transcription. So we then wanted to know if neuronal physiology was impacted in some negative way by dumping in all of these plasmids. And to assay this, we collaborated with Bernardo Sabatini's lab. Genia Kozarovitsky then was performing physiology on the system. And what Genia would do would be electroporate plasmids in utero into the lateral ventricle and drive the plasmids into progenitor cells. The cells that ended up with those plasmids were often the pyramidal cells in the cortex. So she could wait a few weeks and make sections or slides or slices and then do physiology on them. So this is a series of images from those sections. So you can see that there's a lot of cells that don't have GFP. If you look over here in the GFP channel, you can see these two cells are expressing GFP. Here's an example of a cell that's not. And this cell is also expressing TD tomato, implying it's gotten all of the components and the system is working. So the question is, would this TD tomato cell exhibit any kind of aberrant physiology relative to a GFP only or a non-transfected cell? So Genia looked at that in a variety of ways. These are the trains of action potentials from injection of current. So a non-electroporated cell, a GFP only, or a TD tomato cell, and you can see they look quite similar. If she quantitated that, you can see that down here. There was no perturbation of the action potential frequency from the introduction of all of these components. There was no change in the membrane resistance or capacitance. So this then says it is possible to add all of these components and still carry out physiology. Now, we wanted to know if you could use this to do physiology experiments, and for this we collaborated with Botan Roscoe's lab at the FMI in Basel. They record from retinal cells. So we went back to this Gustafson GFP animal that I introduced in the beginning, which has GFP only in a subset of bipolar cells. The connectivity of those bipolar cells is not known. Here's just some images of those cells. So we electroporated in initially just to see if this could drive a gene under control of the system. We introduced this TD tomato, and you can see we only got tomato-positive cells among the GFP-positive cells, so this was working specifically. And then we electroporated a construct which had channel rhodopsin, an optogenetic protein, fused to cherry. Now, many of you know that one can then use light of the right wavelength to depolarize a cell if it includes channel rhodopsin. So we electroporated this plasmid in and used GFP, the endogenous GFP then, to activate the cells. Sorry, this is advancing when it shouldn't have, but Botan's lab then records from ganglion cells, the output cells of the retina, which are connected to the bipolar cells. And when they shined light, on the right wavelength shown here, and they blocked input from photoreceptors, they could then see channel opening very well in the presence of this channel rhodopsin construct, only in the GFP cells, and they did all the proper controls to see that this was indeed under the regulation of the system. They could then inject dye into the cell that they had recorded from to see something about the morphology of the ganglion cell that was being activated by these bipolar cells. The bipolar cells are here in red. So one can start to then put together some concept of what kind of circuitry these bipolar cells in this ganglion cell are participating in. We wanted to know if it would also extend to other species other than mice. There's quite a collection of GFP strains in the zebrafish, and that's shown here. We collaborated with Matt Harris's lab, Catherine Henke in his lab, and we looked at a GFP transgenic of zebrafish, introduced the components, and you can see we could get TD tomato readout. Again, it was very specific. An animal with no GFP had no TD tomato. And this is an animal we were just monitoring for structure and morphology to see, again, if development proceeded normally in the presence of these components. And as far as we can tell, it did. So to summarize the T-dog portion, so T-dog for transcription dependent upon GFP, the system can drive gene expression, and we can use different transcription activation domains, dialing it up or dialing it down, depending on the activation domain. One can use a variety of binding sites. I showed you TET and GAL4, but you can extend it to others. It can be regulated by drugs. I showed you that with the TET system and doxycycline. It can lead to Cre-mediated excision. I didn't show you that, but instead of tomato or channel rhodopsin, the output gene can be Cre, in which case it can then excise floxed alleles or Cre-dependent alleles. I showed you some of the results from Botan's lab. One can use an optogenetic system, and it works across species. So this was well and good, and these plasmids are all available to anybody who wants them from Addgene. And I encourage some of you to think of applications. Again, you can drive any gene. For example, CRISPR could be activated only in GFP-positive cells. But we wanted to see if this system could extend to non-modular proteins. So I told you these transcription domains are very useful as fusion partners because they have these autonomously folding domains that carry out specific functions. But many proteins do not, or they're not known to have such domains. So we wanted to know if we could use a at least known modular domain protein and drive it, again, as fusions that would be GFP dependent. So the system I just told you about had three components. It has a transcription activation domain, a DNA binding domain, and then one has to introduce a plasmid that will be recognized and driven by the system. So 3 components. If one can instead fuse the protein of interest, up here the example is Cre, but if one took Cre, instead of driving it transcriptionally, took the protein itself and fused it to these GBPs, one could reduce the complexity to 2 components and have the protein activity directly regulated from scaffolding on GFP. So this was very hard to get to work, but Jonathan managed to do it. It was not obvious where to fuse these things, how to make these linker structures, where exactly to split the Cre, but he managed to get it to work. There were a couple hundred constructs that went into this. And again, he used luciferase as his readout in vitro and was able to get a system that had a very good GFP dependence. So this is Cre recombination in the presence of GFP or in the absence, and this is without some of the components. So in vitro, it worked quite well. We then wanted to test it in vivo, and as I've shown you, we can use electroporation, but we know that many neuroscientists are interested in using this kind of system with an AAV delivery. AAV vectors are very convenient to inject into all kinds of locations in the entire body. They happen to work quite well in the nervous system. So although we could use electroporation, and we tested what we call Cre dogs for Cre dependent on GFP, and it did work well, we decided to go to a viral delivery system. We've also created some transgenics, which we are currently testing. So if you have a system where you want to turn on the components in all GFP cells, not just those that are infected with the virus, hopefully you can use a transgenic cross. And for fish, they can use microinjection. So we created AAVs that encode the 2 fusion proteins, the 2 components, the 2 halves of the Cre. We have as a readout for this set of experiments, tomato. And then we included GFP or a non-GFP related green fluorescent protein called ZS green. And the question was, when we include GFP but not ZS green, will we see tomato? That would actually then mean that all 4 of these components have to be infected into the same cell. And the efficiency of that just depends on the titer of the virus and the injection site that you're using. So here's now a confocal section taken from a retina when GFP was included or ZS green was included. This is at the level of the horizontal cells. This is a cell type in the middle of the retina. You can see we got very good infection. We have lots of GFP. And we also have lots of the tomato, indicating that the Cre dog was actually doing its job. If we use a non-related protein, a non-GFP derivative, this is just a different protein called ZS green, we got no tomato. So again, the system was very tight, well under 1% in terms of background. But we introduced GFP here with a virus, but we want to be able to see if this system will work with endogenous or transgenically expressed GFP. And for that, we collaborated with the Andy Huberman lab, Ankur Dande, his postdoc. Andy's lab has characterized lines of mice where GFP is expressed in subsets of ganglion cells, these output neurons. And one of them in particular, you can see here, this is a view from the inside of the retina looking at a subset of the ganglion cells that light up with GFP in this transgenic. We then gave them a cocktail of viruses, again, that has the 2 components for Cre dog and TD tomato as a readout, and they infected in vivo with this cocktail. And the question was, would TD tomato turn on only in the GFP positive cells? And that was true. That's shown here, and you can see that only GFP cells turn this on. Now, again, this wasn't super efficient, and that has to do with the viral titer and delivery method. They've studied these a lot. They know where they project in terms of targets in the brain. The dorsolateral nucleus is the target. So they also looked to see if these cells with the TD tomato were still targeting normally and had normal morphology, and at least to a first approximation, they do. So again, hopefully these things are benign or neutral in terms of neuron development and physiology. But to look more closely at physiology, we again wanted to have recordings from slices, and for this we collaborated with Wade Reguer's lab. Stephanie Rudolph, his postdoc, had a transgenic line that expressed GFP in Purkinje cells of the cerebellum and molecular layer interneurons. And so she introduced the CRE-DOG components, and this time the readout was again channel rhodopsin. It wasn't just TD tomato. Here's the injection site, and again she looked at specificity and quantified, and background was again well under 1%. That is, cells that were not GFP positive don't express this channel rhodopsin fusion. She then could do slices and do recordings, the same kinds of things that Genia did, and looking at Purkinje cells or the molecular layer interneurons. And if you look down here, again, you can see membrane properties, action potentials were unperturbed by the introduction of all of these components. So again, hopefully meaning that one can use the system and drive channel rhodopsin or any optogenetic protein without worry about perturbing the physiology of the cells through the method itself. So for CRE-DOG then, the summary is that we can do gain and loss of function. That is, GFP-positive cells basically become CRE-positive cells, enabling cells to then recombine whatever CRE-dependent alleles are present, either by viral delivery or an endogenous CRE-dependent allele. I'm not going to go through this, but there is a way to use viruses, other viruses, to do transsynaptic tracing, and you might know that when one uses those systems, it's very useful to have the viruses for the tracing infect particular cells. Normally that's arranged by a CRE allele in a transgenic. One can now envision using the whole repertoire of GFP-positive transgenics and directing your synaptic tracing to just the GFP-positive cells rather than having to have a CRE or a flip line. I showed you some examples of optogenetics with CRE-DOG, and I'd like to spend a minute talking about this. Many of you probably know, again, that GFP is often expressed in more than one cell type. CRE often as a transgene similarly has that problem, flip and other recombinase. Similarly, we don't get a single cell type from any of these transgenics. Susan Demecky's lab has worked out something called intersectional methods to then take a mouse that has flip under one set of regulatory elements, CRE under another, and mate those two animals such that where a cell expresses both CRE and flip, one can get a more refined subset of cells that one can then manipulate genetically. So if one does that cross and then introduces a gene that's CRE plus flip dependent, one will get activation of that gene only in a subset of the CRE or the flip cells, and that gives you much more specificity for the perturbation or the manipulation. So one can now do that with GFP plus a recombinase line. So one can take any of the GFP transgenics and cross to a CRE or a flip line. Okay, so everything I told you about so far was about GFP nanobodies, using GFP in transgenics to get the cell type specificity and then using the nanobodies to activate genes. But we wanted to know if we could go beyond GFP nanobodies and use other cell type specific proteins, endogenous proteins, non-transgenic proteins, which would give us much more facility, much more access to other cell types that weren't in a transgenic marked fashion and also might go beyond model organisms. So the next part, the last part I'd like to tell you about is Jonathan and Eugene Lansky's idea to expand this to non-GFP nanobodies. So they had the idea to create a conditional system where the condition was the presence or absence of an epitope. So here's the idea, to take a nanobody, mutagenize it so it was unstable in the absence of its cognate epitope. Make fusions of that destabilized nanobody to various moieties, CRE, flip, Cas9, whatever you'd like. And the idea was in the absence of the binding partner, the epitope, it would get degraded, so it would be inactive, no activity of the fusion protein. But in the presence of the epitope, and we started with GFP, it would be stabilized and therefore active. So one could have CRE or flip or Cas9, et cetera, active only in a cell with the epitope. So this is now even a simpler system because it's a single protein that one needs to deliver with whatever the delivery mechanism is. And it's also a generalizable strategy that one can envision binding partners being stabilized through their interaction and being unstable in the absence of. It doesn't even have to be nanobodies. So to see if this would work, the first thing we did is we took the nanobody, one of our favorite ones, it's one of the GFP nanobodies, and we mutagenized it throughout the protein using PCR mutagenesis. We took that library and we fused it to a blue fluorescent protein. So this is not a GFP derivative, this is called tag BFP. It was a readout that we chose. So we have now a library of mutagenized nanobodies fused to BFP. These were then encapsulated, made into a retroviral library. We then took that library and we infected cells in culture And the library not only had the mutagenized fusion proteins, but it also included an HC-RED readout so we could find infected cells regardless of the status of this fusion protein. So we infected cells, and then we faxed out or isolated red cells. So now we know these are the infected cells. And we looked for the ones that were red but not blue. Okay, so that was the key. No blue fluorescent protein, that is, no fusion protein expression. Now that could be for a variety of problems, but we wanted cells that did not have the blue fluorescent protein. The next step was to take those cells, grow them for a bit, and then infect them again, but this time with an AAV encoding GFP. And the hope was that when GFP was present, we would now see blue, because some of these, again, were unstable in the absence of GFP, but could respond to GFP by becoming stable and therefore blue, and indeed we could find those. So this worked reasonably well. We then isolated these individual mutants, sequenced them, and then tested them individually. We made combinations of the mutations, looked for those that were the most destabilizing, and we also tested them in other ways, which I'll tell you about now. But first, just again, our go-to experiment is to see if this works in vivo. We use the retina, and in this case now we're transfecting in or electroporating in a single destabilized nanobody. This again was active against GFP. And then we encode either GFP or an irrelevant protein to see if we get blue only when GFP is present and without the other protein present. So again, if you look here, that worked very nicely. So here's the case where we included GFP, lots of GFP positive cells. And this is pseudocolor, but this is actually blue fluorescence that was turned on only in the GFP positive cells. Very low background. If we didn't include GFP, we only included DS-RED as a control protein. No blue fluorescence. So this was very tight in vivo. We then were curious as to whether it was working with the method that we were hoping, or through the mechanism we were hoping, which was a degradation scenario. So we tested this in vitro. These are Western blots. So we put in our destabilized fusion protein with an epitope that was easy to score on a Western blot. All of these cells had the same amount of that plasmid. YFP is a GFP derivative that should stabilize. So if you look over here, when YFP was present, we had a whopping amount of the fusion protein, shown here. If YFP was absent, we put in the same amount of the fusion protein. We had no fusion protein. So there's a huge difference in the amount of protein present when epitope is present, consistent with the idea of degradation. We can also titrate in the epitope shown, shown here. This is the epitope, but here's the fusion protein now stabilized. And this is actually a fairly linear response, which is nice in terms of controlling what's going on just by changing the dosing of the epitope. I'm going to skip over this for time, but it's nice to have a linear readout. If we have a ternary or a binary system, you can actually titrate out the interactions for those of you who are aficionados of this sort of thing. But now we also want to know, is it working through the ubiquitin proteasome system for degradation, which was our guess? And to test that, we did the experiments again in vitro, and we included blockers of degradation through the UPS system. So look over here. This is the destabilized fusion protein. When it is delivered without its epitope and with an active UPS system, we get no fusion protein. If, on the other hand, we include either of these two blockers of UPS, we now get the protein back. So this is all consistent with the mechanism we envisioned, that in the absence of epitope, the protein is being degraded through the UPS system. Okay, now looking at the sequences, these mutations that then gave us the conditional stability, we were happy to see that these mutations were in the framework or the heavily conserved regions of these camelid nanobodies. So these are 3 different species of nanobodies, and what we're showing here is the framework regions, which are the heavily conserved regions among all nanobodies, and this is where these mutations mapped. So these are the most important of the mutations we found. So this was great because what it told us is we might be able to just substitute these mutations in other nanobodies without having to do the laborious screening procedure. We could just take any nanobody whose sequence was published, swap in these mutations, and see if we now had conditional stability, and we could test that by adding the epitope. That worked quite well. The majority of nanobodies we tested were conditionally stabilized by these mutations. And I'll show you some of the applications then that we've put this to. So I'd like to just show you this set of panels. This was now to make a flip recombinase that's dependent on GFP. We used our destabilized GFP nanobody. And look at the activity over here. This is, again, a readout for luciferase after flip activity. We took flip and we fused it to either a single destabilized nanobody or a double. And we asked, is there GFP dependence for recombination? And if you see over here, we have a lot more recombination when GFP was present versus when it was absent. So this is about a 600-fold induction of flip activity by having this dual nanobody domain, and again, having these be destabilized domains. If we don't use the destabilized domain, here's the normal nanobody. It doesn't have GFP dependence for activity. Now, this was great because, first of all, it gave us this reagent, FlipDog, which, again, you can all use as in the same way you use CreDog. You only have to deliver one protein. It's active. It can be used, again, in a combination with, let's say, a Cre strain. If you have a Cre plus a Flip mouse cross and you introduce this, you'll have, again, a Cre plus Flip intersection for your genetic manipulation. The second thing this showed us, though, is that we could put 2 nanobody domains on a single protein, and that gave us the possibility of doing an AND function, again, to create more specificity in the flip readout. So we had nanobodies to a number of different proteins. I'll just show you one example over here. We had a nanobody from the literature for an HIV capsid protein. We introduced the conserved mutations, and we found antigen dependence. We then took that destabilized anti-HIV domain and fused it in tandem with our anti-GFP destabilized domain, again fused to flip. And we asked, is flip activity stimulated only when both antigens are present? And indeed that worked. That's this column over here. Again, these are in vitro readouts of luciferase. If both antigens are present, we have a great deal of induction. If none of the antigens are present, we have almost no activity. If one is present, we have a little bit of activity. So this then opens up the possibility, again, to gain access to a more specific subsets of cells by using the combination of epitopes to turn on a dual nanobody fusion protein. So this is the last application I'll show you then. This is now to use these single nanobody fusions that are dependent upon the presence of epitope and to use them as detectors. Now, this is useful in a number of applications. If you just want to know if your antigen is present in a cell, let's say you're working in a system where you're looking at induction of an endogenous protein, one of these introduced intracellularly will give you the ability to see the turn on of your protein. This works much better than just putting a fusion protein in, because if you put a fusion protein in that was not destabilized, so let's say you had a nanobody to antigen X fused to GFP or BFP, that nanobody is stable in the absence of epitope, so your cell will have sort of a blue haze as a background. Here, if you put in a destabilized nanobody, there's no background. The nanobody goes away unless the epitope is present. So this is a very sensitive detector system. We wanted to test this on a virally infected set of cells, and we went for an HIV. We worked with Jonathan Lee and his postdoc, Bazad Etamad. And what we wanted to know is whether HIV capsid protein, when induced, would enable cells to turn red if we put in our fusion protein, our destabilized fusion protein, to detect that. So we took a set of cells that were the parental line. These are uninfected, so there should be no HIV epitope, and therefore no red from the fusion protein. That parental cell line is infected with HIV to create a derivative line. HIV in this scenario is latent. So you probably know that HIV, once it infects cells, can go quiet, not express any of its genes. But for reasons that are unknown in a person that's chronically infected, they will reactivate the genome. And that's a point of study. People who are studying HIV biology would like to know what activates. And this gives them a way, if they can study this with our fusion proteins, this gives them a way to screen for activation in a live cell population. They don't have to permeabilize the cells or do any of the other things that you have to do normally to see an epitope turn on, especially if it's an intracellular epitope like the HIV capsid protein. So this cell line has a quiescent genome, but that genome can be activated by a chemical called PMA. So we took the parental cell line, the non-reactivated cells, and the reactivated cells. We introduced our destabilized anti-capsid nanobody, tagged in this case with RFP. This is just a transfection-controlled GFP. It doesn't have anything to do with the nanobody. We then introduced these constructs and then faxed to ask, would we get some red cells if we reactivated the HIV genome? And you can see from the fax plot, this is the area where the red protein was seen among the GFP transfected cells. The parental cells or the non-reactivated cells did not have any red cells. That's quantified over here. This is the reactivated HIV-infected cells. This is the parental line. This is the non-reactivated infected cells, and again, the parental cells. So this is a very sensitive way then to detect reactivation of HIV, and it can be used in high throughput screens. It's similar, it can be used to detect viral antigen and other infection scenarios. It also can be used, again, to look at the induction of an endogenous protein, if that's your interest. And this is the last experiment then. We wanted to know if we could induce Cas9. I'm sure most of you know about CRISPR. One can then get genome editing specifically in the cell that expresses the epitope. We again used our HIV destabilized anti-HIV nanobody in a double fusion scenario with Cas9, the enzyme. We tested this in a tissue culture system where LAC-Z would be turned on. The guide RNA was then directing the Cas9 to excise a transcriptional stop. So Cas9 activity is indicated by LAC-Z activity. We looked at a wild-type normal allele of Cas9 in this assay, and it worked quite well. So this is now LAC-Z readout. So this is the normal, non-modified Cas9 in the presence of guide RNA. We set that activity to 100%. In the absence of guide RNA, there's no activity. And this is our destabilized fusion protein over here in the presence of HIV capsid. You can see we got quite a bit of activation, about 70% of what we see in the normal wild-type Cas9 allele, and it's dependent upon epitope. If we leave out HIV, we get very little activity. So now one can then use these fusions to direct the genome editing. I think this could even be used in a scenario where one is trying to affect, let's say, deletion of a dominant negative allele for human therapy. And I think this latter bit opens up the possibility that if we have nanobodies for endogenous proteins, one can introduce them into animals which are not our standard model organisms. These are just some of the organisms where actually there are transgenics available that shows us at least that we can introduce exogenous gene. But as long as one can use viral delivery systems, one can then think about developing nanobodies with these activities, for example, to target GABAergic neurons in the cortex or whatever one is interested in. And I'll just end by thanking all the collaborators. I think I mentioned this as I went along. I would like to just point out one other thing, and that is I know that sometimes people are intimidated by a lot of molecular biology. You neuroscientists are extremely sophisticated people. You do not need to be intimidated by any of this. And in fact, if you like, you can just buy these AAVs and inject them. You don't need to build any of the constructs or really work with anything that might seem complicated. And you can buy them from Luc Vandenberg, who has the AAVs produced at the Mass Eye and Ear Virus Core. It's a nonprofit core. And please contact me if you're also interested. So I'll stop there and be glad to take questions.
We have 5 minutes for questions. Go ahead, Greg. Wait 1 second.
Thank you for the talk. I was wondering if you had tested your system on GFP lines where GFP is produced as a fusion protein with the membrane or cytoplasmic protein.
Yes, we have. So we've used a fusion protein that directs GFP to the nucleus, and it works well with that. So I think nuclear and cytoplasmic proteins are fine. We've used a lot of cytoplasmic proteins. Membrane proteins, I think it's going to depend on where is the epitope and how does the nanobody interact with the epitope. Does it do it before membrane insertion? Probably not. So my guess is it's not going to work on membrane proteins. Sorry if you have one of those. You have to come up with another strategy.
I'll ask, can you hear me? I'll ask 2 questions. Yeah, sure. Technical question. So I think you did a beautiful job of acknowledging my introduction to you. You know, incredibly innovative and sophisticated methodologies.
I think Jonathan gets that credit.
Yeah.
Not me.
All right. Well, you guided him. The 2 questions are first about the AAVs, and obviously for the use of them in vivo for the types of work a lot of us in mice want to do, the efficiency is really important. So is it serotype dependent, or if we buy the AAVs from the Mass Eye and Ear, is it 90% efficient getting 2 AAVs into 1 cell, 80%? I know but what's your experience or opinion?
So yeah, again, most of my experience comes from the retina.
Yeah.
So if we introduce an AAV that encodes GFP and an AAV that encodes TD tomato, and if we inject the subretinal space at postnatal day 0, we have virtually 100% co-infection. and that's with 2 different serotypes. But I think that extrapolates differently to I think it really depends on your injection site. It really depends on the titer. I think the destabilized antibody is a better situation because you don't have to get co-infection with 2 CRE-dog components. You can just go for the one, and again, flip-dog is a single component. But nonetheless, we've given these components to a number of people, and they report exactly what you would expect. It depends on their ability to target the GFP cells at their injection site and the titer of the virus. So I think it's going to come down to that. But I'll also mention we did make transgenics. So if someone wants to have a system where they can get more of their GFP cells with the components, they can breed. The caveat with that is going to be, when do you want to turn on the system? Will you get any kind of temporal control from that breeding scenario? And again, it's going to depend on when your GFP comes on.
You answered my second question. I was going to ask you about the transgenics. So these are presumably transgenics where the components for, let's say, the CRE dog are ubiquitously expressed.
Theoretically, we put them into actually Leach-von-Loew's locus H11, which should be ubiquitous.
Okay.
During your opening, you said that your passion was actually about development. And so I was wondering, given, and I'm a person who thinks about vision and the retina and so forth, and I'm wondering, given the techniques that you've described, what would a question about development and about visual development that would matter for vision that would now be open to us to explore with the techniques that you develop?
Yeah, it's a good question. So I think that it's really a matter of genetic access, that there are GFP lines that exist in the retina as well as elsewhere that we can now say, okay, we want to target those GFP cells and turn on or off genes that we couldn't access before in a specific way. Or we can also do, we're working on transsynaptic tracing. We can also ask, and as I showed you an example, of GFP positive cells, subsets of cells, which we can now ask questions in terms of circuitry and physiology. So it's really just an expansion of things that we could do before, but it's now expanded in terms of our ability to target specific cell types. So for us, it's really access now to a much wider range of cell types for genetic access.
Last chance. Okay, let's thank Connie once again.
For more, please visit us at stanford.edu.